Hello and welcome to February's Longevity Review, where we will take a look at how the combination of omega-3, vitamin D, and resistance exercise can slow down the pace of aging in humans, how young blood plasma can help with surgical outcomes in a human trial, how Klotho gene therapy can increase the lifespan of mice, and in the Canadian content study, how cellular reprogramming can protect retinal neurons and maintain vision in a mouse model of multiple sclerosis. My name is Chris Linnell, and I am the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do and how you can support us, then please check out the links below this video, where you can also find links to the studies being reviewed here. In addition, don't forget to like this video and subscribe to our channel. So let's take a look at the first study. As we await the introduction of truly effective longevity treatments, what can we do in the here and now to help us reach that time point? This study might provide us with a partial answer to that question. So this study was a subset analysis of the larger Do Health trial, which was an intervention study in over 2,000 healthy European adults over the age of 70. They gave them 3 treatments, either individually or in combination, either 2,000 units of vitamin D, 1 gram of omega-3, or a home exercise program, which consisted of 3 30-minute resistance exercise sessions per week. Looking at the allocation of participants, we can see what the 8 different arms of the trial were. In this subset analysis, all of the participants were from Switzerland, and there were 777 in total. 95 of them had no treatment, 101 only took vitamin D, 98 only took omega-3, 92 only did the home exercise program, 95 took vitamin D and omega-3, 104 took vitamin D and did exercise, 95 took omega-3 and did exercise, while 97 did all 3 treatments. Before we look into the details of this study, we will briefly look at the results of 4 prior studies using the Do Health data. The first study looked at blood pressure, fracture risk, physical performance, infections, and cognition. And although for most of the parameters they did not find any significant difference, for infections, they found that those who took omega-3s had a 13% lower risk of infection, which was particularly significant for upper respiratory tract infections and urinary tract infections. In the second study, which looked at fall prevention, they found that once again, only those who took omega-3 had a reduction in fall risk, in this case, 10%. The third study looked at cancer risk, and here the individual treatments resulted in a 24 to 30% reduction in cancer risk. The pairwise combinations resulted in a 44 to 48% reduction, while all 3 treatments together resulted in a 61% reduction in cancer, which is quite considerable. The final study was on preventing the transition to pre-frailty. And here, once again, they found that when the 3 treatments were taken together, there was a 39% reduction in the transition to pre-frailty. Links to all 4 of those studies can also be found below this video. Now, moving back to the present study, this study looked at how the biological rate of aging changed over the course of 3 years using 4 aging clocks: PhenoAge, GrimAge 2, and Dunedin Pace. And we'll look at this graphical abstract to give a brief overview of those 4 clocks. So starting with PhenoAge at the top left here, this uses 9 blood chemistry markers for its analysis. And in fact, this is something you could test yourself using standard blood work. Calculators for this can be found online. Next, GrimAge 2 uses 9 plasma protein markers as well as smoking history for its analysis. The Dunedin Pace uses data from the Dunedin longitudinal study and shows how the rate of aging changed using 19 biomarkers of organ system integrity. The last GrimAge uses 7 plasma protein markers, which are listed here. They are all related to diseases and dysfunction. I will explain what they relate to shortly once we look at some results. And here are the results for the 4 aging clocks, starting with PhenoAge in the top left. And as you can see, here are the 7 treatment groups on the left-hand side. And any results to the left of this dashed line indicate a reduction in aging. Anything to the right is an acceleration. Out of the 3 individual treatments, omega-3s performed the best, as you can see with this black square being furthest to the left. The pairwise combinations showed an improvement, with once again those containing omega-3 performing the best. However, the best result overall was when all three treatments were combined, as you can see with this black square being furthest to the left. So this is indicating that there was an additive effect with combining these treatments. For Grimage 2, only omega-3 on its own showed a significant effect, as can be seen by the fact that its error bar, which is this black line here, was fully to the left of the dashed line, whereas all the others also went over to the right-hand side. For Grimage in the top right, once again, only omega-3 showed the greatest reduction. Also here, its error bar went over the dashed line to the right-hand side. Likewise, with the combinations, only those containing omega-3 had the black squares on the left-hand side. The same went with Dunedin paste, with only omega-3 on its own showing a significant reduction. And once again, the combinations containing omega-3 were the ones which performed best, though there wasn't an additive effect compared to omega-3 on its own. What did these results mean in real-world terms? Well, according to the authors, the combined intervention resulted in a 2.9 to 3.8 month reduction in biological aging over the course of those 3 years. Now, that doesn't seem like much. However, if we extrapolate that over a longer period of time, over the course of a decade, it would mean being roughly 1 year younger in biological aging terms. Now, looking in more detail at those 7 plasma protein markers from Grimage 1, once again with the 8 intervention arms on the left-hand side and the dashed line in the middle here showing a reduction on the left and a worsening on the right. Again, lower is better for all 7 of these. We start with GDF-15, which is a marker for cardiovascular disease, death, and heart failure. And here we can see that omega-3 performs slightly better than the other 2. And there seemed to be an additive effect with the 3 pairwise combinations doing slightly better, once again with omega-3 performing best. However, all 3 treatments together performed best of all, as you can see by the black square being furthest to the left. PAI-1 is a marker for cancer, atherosclerosis, and other diseases. And here we see yet again that omega-3 performed best out of the individual treatments, as it did in the pairwise combinations. However, the triple combo did not improve on that. TIMP-1, which is a marker for cancer, yet again, omega-3 performed best in the individual treatments, and best overall was omega-3 combined with exercise. Moving on to B2M, which is another cancer marker, omega-3 performed best, and I think you can see a trend emerging here. However, for this protein, the triple combo performed best of all. ADM, which is a marker for vascular disease, omega-3 was once more at the forefront for the individual treatments, while the combination of omega-3 and exercise performed best in the pairwise combinations. However, the triple combo didn't seem to do as well. Looking at leptin, which is a marker for obesity and metabolic disorders, yet again, omega-3 on its own and omega-3 with exercise performed best. And finishing off with cystatin C, which is a marker for kidney disease, none of the treatment arms made a significant difference, with perhaps omega-3 combined with vitamin D performing slightly the best. And there we have the results of this study. Now, they weren't particularly groundbreaking, and by taking these supplements and doing exercise, you're not going to live to 100. They did, however, show that these treatments could make a small difference, and over the course of decades, that small difference could rack up. One thing to bear in mind was that the participants in this study were healthier than average. They had lower levels of vitamin D deficiency and had higher levels of physical activity, with 88% of them being considered physically active. And as we have seen in other trials, and especially in mice, animals which are already healthy show less of an improvement. So we can go out on a limb and hypothesize that for the general population, this intervention would show greater effects. All in all, this was a useful study, and I particularly like the fact that it was done over 3 years. Too many studies I read about are only for a few weeks or a few months at best. Though, of course, it would be even better if we could see even longer studies. Considering the ease as well as the low cost of this intervention, and also taking into account that there were no adverse effects, I don't think it would hurt if we all supplemented with vitamin D and omega-3. And we all already know that exercise is good for us. For those of you living in Canada, considering our high latitude, supplementing with vitamin D in the winter is especially important. On to the next study. Ever since the first heterochronic parabiosis experiments took place more than 2 decades ago, whereby an old and a young mouse were surgically co-joined and the old mouse became younger while the young mouse became older, the idea has emerged that there are youthful factors circulating in young blood which could potentially rejuvenate older animals. Since then, numerous studies have taken place in mice which have proven this to be true. However, data in humans has been sorely lacking, until now at least. In this study, researchers sought to see what would happen if they gave older adults who were undergoing major joint replacement surgery young plasma, and whether or not that would change the proteomic profile and speed up their recovery. Let's start by taking a look at the study design. So first off, I should mention that this was a small study. Looking at B on the bottom left here, you can see that there were 18 patients in the treatment group and 18 placebo. The treatment, called GRF 6021, was a 5% plasma fraction, whereas the placebo was a saline solution. Both groups were given 4 infusions: the day before the surgery itself, just after surgery, and the day after surgery. Blood samples for analysis were collected on those days as well, and the patients were followed for 6 weeks afterwards. Looking at the first set of plasma proteomic results, we can see here a visual representation of the 6,596 proteins which were analyzed. Now, I won't go into this, but it does seem quite comprehensive. Of more interest are the differences between the treatment and placebo groups, which we can see down here. What we have here is the differences between the 2 treatment groups in their proteomic profile. On the day before surgery, the day of the surgery before the surgery itself, on that same day after the surgery, and then the first day after surgery. And whereas the profiles were quite similar the day before surgery and before the surgery itself, they diverged quite considerably after the surgery and the next day. This can also be seen in these graphs below, whereby the black line is quite close to the red dotted line the day before and the day of before the surgery. The black line is further away from the red dotted line after the surgery and the next day. What some of those differences are can be seen in C below. The 12 pathways detailed here all deal with either inflammation or are associated with cancer. And you can see that before the surgery, they are not that much different. However, afterwards, and especially on the first day after, major differences emerge. Now, the authors aren't specific about this, but I am assuming that the pathways are higher in the placebo group. Next, the authors looked at the immune response. And on the left here, you can see all the various different immune cells. On the right here, we can see the activity levels of 2 proteins across those immune cells. On the bottom, NFKB is an inflammatory molecule, and it did not show any real change in activity. So lighter colors show greater activity. However, another molecule, STAT3, which elevated levels can lead to greater cognitive decline after surgery, there was increased activity over the course of the first day, as can be seen by the lighter shading on day 1 post-surgery. Looking at the overall differences here, with once again B being the day of the surgery but before the surgery, C the day of the surgery but after surgery, and D being the next day, there was no significant difference between the 2 groups before surgery, as can be seen by these 2 bar graphs, and the fact that the black line was fairly close to the red dotted line. Immediately after surgery and the next day, significant differences once again emerged, as can be seen by the differences between the 2 bars and the fact that the black line is quite diverged from the red dotted line. Next, they looked at specific immune cells, and in those immune cells, 2 proteins stood out from the rest, and they were PERK in Treg cells and Treg memory cells. PERK is associated with tumors and Parkinson's, and blocking PERK can be neuroprotective, so lower levels are better. The green bars indicate the treated patients, and the light blue bars are the placebo. And as you can see, levels of PERK in both these cells were much lower. On the day after surgery, 7 proteins in different cell types stood out. And the first 3 here involved STAT3, and as I mentioned before, STAT3 is associated with cognitive decline after surgery. So lower levels in these 3 cell types are a good sign. MAPKAPK2 is associated with tumor progression, so lower levels are also beneficial, and they were indeed lower in the treated group. IKB blocks an inflammatory pathway, so higher levels are beneficial, and it's good to see that that was the case here. CD8 is crucial for robust immune protection against infection, so higher levels are beneficial, and that is indeed the case here. As for non-classical monocytes on the right here, they can lead to greater inflammation, so lower levels are better, and once again, that is the case here. Lastly, when looking at the immune system, changes in STAT3 were mainly found in naive CD8 T cells here on the left, which are part of the adaptive immune system, whereas the other three on the right here are part of the innate immune system. So all in all, it appears that young plasma can dampen down the immune and inflammatory response to surgery, which should be a good thing. However, how does this impact patient outcomes, which is the most important point? Now, although the authors talked about it in the text, for some reason they decided to hide the figures in the supplementary section, which seems strange to me, as I think these are the most important results. However, I think this is due to the fact that none of these results were statistically significant, as there was such a small number of participants in this trial. However, what they found was, starting on the left here with fatigue and function, which is the time it took to reach 50% recovery in days. For the placebo group in gray here, it took 16 days to reach that time point, whereas for the treated group in green, it only took 12 days. In terms of the amount of time it took to reach only mild pain, the placebo group took 18 days on average, whereas the treated group took only 12 days again. In terms of opioid consumption, which is also obviously an indicator of pain, there was no difference over the course of 6 weeks, as can be seen by the black line here being pretty much the same. However, when it came to the 2 days they were in hospital, there was a difference. On the first day, the placebo group consumed 3.4 grams, whereas the treated group only consumed 2.3 grams. On the second day, the placebo group consumed 2.3 grams, whereas the treated group only consumed 1.9 grams. Lastly, in terms of the function of the operated leg, or the time to mild impairment, there was no difference between the two groups. So what does this all mean? Well, firstly, this was a small pilot study, so these results are very preliminary. However, they do align with the mouse studies, showing that young plasma can reduce inflammation, which clearly has benefits in terms of surgical outcomes. There are two other things worth bearing in mind. Firstly, and the authors don't really mention this in the text, but the placebo group was considerably younger than the treated group. I mean, they were randomly divided, and the average age of the placebo group was 64, whereas the average age of the treated group was 71, so a 7-year difference. And that could make a quite considerable difference in terms of inflammatory response due to surgery. So the differences might have been greater had they been the exact same age. Secondly, was how young was this young plasma? The average age of the donors was 35, which is not that young when you come to think about it. Mouse studies have shown that the younger the plasma, the better the results, with the best results coming from umbilical cord plasma, which is pretty much as young as you can get. So the question arises, would these results have been even better had the plasma come from much younger people? Indeed, it's intriguing to think what the results would have been had they used umbilical cord plasma. However, this is just nitpicking. It's great to see results in humans as opposed to mice, and one can only hope that there will soon be many further studies using young plasma in humans. Based on all the results in mice, this is an area I'm very excited about. Moving to the third study, Klotho was one of the first longevity genes found in mice back in the 1990s, whereby increasing its expression lengthened mouse lifespan, while knocking it out reduced it. Its name comes from Greek mythology, where Klotho was one of the 3 fates which governed our lifespan. She was the one who spun out the thread of life. In humans, lower levels of Klotho have been linked to various diseases, including kidney disease and increased mortality. Klotho comes in 3 forms, and it is the secreted form which we are looking at here. If ingested, it gets broken down in our stomach, and it is too big a protein to cross the blood-brain barrier. Therefore, at present, gene therapy is the only way to increase its levels, and that is what is employed here. This study was the first to test a single injection of Klotho gene therapy in mice on their lifespan. So let's take a look at what they found. Starting with the study design, 96 mice were used, both male and female, and they were divided into 3 groups. They were aged naturally, and at 6 months, 2 of the groups were injected, one with a sham injection of gene therapy, and the other one with Klotho using a viral vector. 6 months is the equivalent of a human in their 20s. The third group were injected with Klotho gene therapy at 12 months, which is the equivalent of someone in the early 40s. They were then tested at 24 months before living out their remaining lifespan. 24 months is the equivalent of a human in their late 60s. In all of these figures, black represents the control, light blue represents getting Klotho at 6 months, and dark blue at 12 months. Looking at the results, starting with body weight in B, with the dotted line showing 6 months and 12 months when they were given Klotho. And the control mice weight peaked at 14 months. The 6-month Klotho mice peaked at 15 months, and the 12-month Klotho mice peaked at 20 months and seemed to maintain their weight much better. In terms of median survival, the control mice lived for 24.6 months. The 6-month Klotho mice lived for 25.8 months, while the 12-month mice lived for a median of 28.3 months, which was a 15% increase over the control. And you can see the survival curves on the right here, with the dark blue line being pushed out quite a bit further to the right. Now, the authors also talked about total longevity, which I think refers to average as opposed to median. And here, the control mice lived to 26.3 months on average. The 6-month mice lived to 29.8 months, and the 12-month mice lived to 31.8 months, which is a 19.7 increase compared to the control. Now, these lifespan results are only in the male mice because the female mice had a very high incidence of ulcerative dermatitis as well as anal bleeding, and a lot of them had to be euthanized. This was seen across all 3 groups, and the authors think that this was not aging related. Therefore, these results sort of weren't incorporated, and therefore they couldn't calculate the lifespan of the female mice, unfortunately. However, the other tests were done in both sexes of mice. Next, they looked at how much Klotho was increased in the mice, starting with DNA expression in the liver. And as you can see, both in male and in female mice, levels increased quite considerably, especially in the 12-month mice. The same was seen with circulating levels in the blood, with once again the 12-month mice showing the highest levels. Also, it appears that Klotho increased more in the male mice as opposed to the female mice, as can be seen by the higher dark blue bars here and here compared to the female ones. Next, they tested physical function. Unfortunately, this is a journal pre-proof, so some of the headings are blocked off, but this has been peer-reviewed. It just needs some copy editing done and the layout sorted out. And they started off with a rotarod test in A, with male on the left and female mice on the right. And a rotarod test is when mice are placed on a rotating rod, which gradually increases in speed, and then the speed at which they fall off is measured. And these levels are rotations per minute. There was a slight increase for the male mice, though this did not reach significance. However, it did reach significance for the female mice, especially the 6-month mice. The second test in B was a horizontal bar where mice were left hanging with their forelimbs off a small beam, and they were timed to see how long it took before they fell off. Here, both the male and female mice saw an improvement, especially the 12-month-old mice, which reached significance, as can be seen by the star on top here, with more stars meeting greater statistical significance. Next, they looked at grip strength, where mice are sort of cling onto a wire while their tails are pulled, and the force at which they let go is measured. Now, this one is a little bit confusing. They were tested 3 times, and in the text, the authors say that the male mice saw a significant increase, but I can't really see it from the bars here. I mean, for the 6-month mice, there's an increase denoted by these sort of 3 stars up top here, but the bars are lower than the gray control bar, so I'm not sure what they're really talking about here. For the female mice, only in the first test did the 6-month mice surpass that of the controls. So, as I say, a little bit of a discrepancy between the text and this figure. So maybe that will be sorted out in the final version. Then they looked at muscle fiber size from 24-month-old mice. So a subset of the mice at 24 months were euthanized to be analyzed further. So here you can see the cross sections, and according to the bar chart on the right here, there was an increase in fiber size, though it did not reach significance.
However, looking at fibrosis, denoted by the red staining in these cross sections here, for the 12-month-old mice, there was a significant reduction in fibrosis. Next, they looked at muscle regeneration. And they took a muscle tissue sample from 24-month mice and grafted them into 3-month-old mice, which have much greater regenerative potential. And you can see a cross-section of those muscle grafts here at the top, and the eyeball test seems to show that the 6 and 12-month-old mice had a greater number of muscle cells than the control mice after a few weeks. And in terms of the muscle fiber size here in B, both the 6 and 12-month-old mice had greater muscle fiber sizes, with the 6-month-old mice performing best. The control mice had a lower peak than either the 6-month-old mice or the 12-month-old mice, as you can see here. Then they looked at markers of cellular proliferation, with Ki67 being that marker. And as you can see in two different tissues, and Ki67 is such a marker, and connected with two other sort of markers, levels of Ki67 increased in both sets of Klotho given mice, indicating that Klotho gives much greater regenerative potential. Next, they looked at Klotho's effect on bones. And here behind this banner, we have mineral density, bone volume, bone perimeter, cross-sectional thickness, bone volume to tissue volume ratio, trabecular number, trabecular thickness, and trabecular space. Now, trabecular is the thicker end of our longer bones. If you sort of imagine a bone, it's sort of thin and then bulges on both ends. Those bulges are the trabecular segment of the bone. And although a lot of these did not reach significance, there did seem to be an overall improvement, especially for the 6-month-old female mice. Now, female mice, just like humans, suffer greater bone loss than male mice with age. The improvement can be better seen with these images here, showing an internal 3D image. And you can see here with the control mice how the female mice had much sort of less bone than the male ones. However, the 6-month-old female mice and the 12-month-old male mice had much higher levels of bone. Looking at the expressions of genes associated with bone growth, there was an improvement compared to the controls, with a greater improvement in females. Only FGF23 saw a decline in females. Then they looked at the brain, or more specifically, the hippocampus. And once again, the banner blocks it off, but on the left here, in these cross sections of the hippocampus, we have the control mice on the left, the 6-month mice in the middle, and the 12-month mice on the right-hand side. And in these bar graphs, it's the men on the left and the females on the right once again. Now, the authors don't really sort of explain this apart from saying that these are all cellular markers important for the correct function of the central nervous system. And as you can see, especially for the 12-month mice, they all had higher levels of these markers than the controls, though this does not appear to be the case with the 6-month-old mice. The same goes with the second set of markers here in B, though not to the same extent. They then looked at neurogenesis. And while levels of that proliferative marker Ki67 wasn't higher, DCX, which are immature neurons, increased in the 12-month-old mice. And the thickness of the granular cells layer was higher in both sets of Klotho mice, though this did not reach significance. Finally, they looked at the transcriptomic profile in the hippocampus, and they found 3 distinct clusters of variants between green 5-month-old control mice, old control mice, and old Klotho-treated mice. And looking at this Venn diagram on the right, which shows differentially expressed genes between either young control mice, old control mice, or young control mice and old Klotho-given mice, there was a much greater number of differentially expressed genes between the young and old control mice compared to the young control mice and the old Klotho-given mice, which indicates that the old Klotho-given mice were much closer to the young control mice in terms of gene expression. Lastly, they looked at what those different pathways were, and they found that compared to the control mice, the Klotho-treated animals presented increased induction of phagocytosis. So that's immune cells which engulf sort of bacteria and viruses, increased immune cell activation, and a decrease in pro-inflammatory signals. Finishing off with the graphical abstract to summarize, we saw increased longevity, up to 20% in the Klotho given mice, enhanced physical condition, I guess that's more physical function. If the mice look better, they didn't mention it in the text or provide any photos. You can see, I guess, the hanging beam test here, and so showing improved muscle regenerative capacity, improved bone microstructure, improved histological markers in the central nervous system, reduced inflammation signaling, and promotion of phagocytosis. Now, a few last things to mention. For some of the tests, you might have noticed that the 12-month-old mice performed better than the 6-month-old mice. And the authors think that as Klotho is more expressed in the liver, and the liver has a sort of a cellular turnover, by the time the mice were tested at 24 months, there was less Klotho in their systems, which actually the serum Klotho levels seem to indicate that as well. Secondly, they obviously need to retest longevity in female mice, and hopefully they won't have those sort of disease issues they had with them. It shows the sometimes random nature of experiments where weird things can happen. They should also test out what happens when Klotho gene therapy is given to even older mice, as the 12-month-old Klotho mice are only the equivalent of humans in their early 40s. The question is, what effects would it have if it were given to, say, 24-month-old mice, the equivalent of humans in their late 60s? But this is a very intriguing result. As I mentioned at the beginning, this was the first time that a single injection of Klotho gene therapy was given to normal mice. And it's great to see the significant increase in life and health span, especially as Klotho gene therapy is hopefully soon to be tested out in humans. Doing a bit of research on the web, I found that a company called Klotho Neurosciences is hoping to start human testing in the fourth quarter of 2025. So with a bit of luck, we might get some human results fairly soon. And finally, the Canadian content study. Multiple sclerosis is an autoimmune disease whereby our immune system attacks the central nervous system, causing issues with motor control and vision. Although not technically an aging disease, since it starts when people are in their 20s or 30s, the cells affected show some of the hallmarks of aging and senescence. So it would be interesting to see if some of the techniques used against aging could also target this disease. In this study from the lab of Alison Fournier of McGill University, they used a mouse model of multiple sclerosis called experimental autoimmune encephalomyelitis to see if retinal ganglion cells, which are the neuronal cells which transmit the visual information from the eye to the brain, show some of the hallmarks of aging and senescence, and if they were rejuvenated via cellular reprogramming, whether that would improve symptoms of the disease. So let's dive into the results. So first, they looked at the genetic correlation between naturally aged mice and the experimental autoimmune encephalomyelitis mice, or EAE, which I'll call them from now on. And in A in the top left here, they found significant correlation by the fact that this line sort of goes at a 45-degree angle, suggesting that there was an aging signature. And in terms of the genes which were up or down regulated, there was considerable overlap between the 6-month-old aged mice and the EAE mice. You can sort of see by these numbers here, 376 upregulated, 116 downregulated, which were more than 50% of the up and down regulated genes in the EAE mice. In C here, looking at the top 100 upregulated genes, there was considerable similarity between the EAE mice at the bottom here and the naturally aged 6-month-old mice at the top. Similarly, looking at a senescent gene set, there was similarity between the EAE mice and 12-month-old mice in F and 6-month-old mice in G. And this was also seen in the motor neurons of the EAE mice. I should also mention that apart from H, all the data is from retinal ganglion cells, or RGCs, which I'll call them from now on. Next, and I won't go into the details here, they looked at genetic pathways which were associated with senescence, and found, to quote, "Together these analyses provide further evidence for alterations in senescence-associated processes in RGCs, indicating that the well-known aging process of senescence plays a role here." Next, they analyzed cells taken from multiple sclerosis patients and found, to quote once again, that the data suggests that in multiple sclerosis, similar to the EAE mice, neurons demonstrate enrichment of genes related to senescence and alterations of pathways relevant to the process of aging and senescence, including DNA damage, epigenetic modifications, and mitochondrial dysfunction. Now, this is a quite detailed slide here, but just to show a couple parts of it. So you can see the cell cycle and senescence. Control is blue, red is multiple sclerosis, and a lot of the pathways for, again, cell cycle and senescence are enriched in the multiple sclerosis patients as opposed to the controls. And similarly, down in H here, you can see where DNA damage is involved. A lot of the pathways are enriched in these multiple sclerosis patients, again, indicating that aging and senescence plays a role in multiple sclerosis. Next, going back to mice and their retinal ganglion cells, they found higher levels of a marker for double strand breaks, which is when both strands of our DNA are cut in half, and an obvious marker of damage, were much higher in the EAE mice compared to the controls, as you can see by these red bars here compared to the, well, no blue bar here and the small blue bar in F here. So onset and peak refers to the fact that multiple sclerosis often comes in waves. It is not sort of a continuous process normally. And onset is the beginning of such an attack, and the peak is obviously when it is at its worst. The difference between C and F is simply the resolution of the microscope used. So F had a greater resolution, so saw more detail, so even picked up small lesions in multiple sclerosis. On the left are all the images from the microscope. In the second from bottom row, it shows a marker of gene transcription repression. So gene transcription is when an RNA copy of the genetic information in our DNA is made, and that then is sent to the ribosome, so proteins are formed. And if that is repressed, then obviously the cell can't sort of produce proteins as efficiently. And here there was greater repression in the EAE mice compared to the controls, as you can see by the red bars compared to the blue. And finally, at the bottom here, they looked at the nuclear envelope, which is obviously the envelope surrounding the nucleus of cells, which is where most of our DNA is kept. And that can become abnormal or misshapen with age. Once again, the EAE mice showed greater abnormality compared to the control mice. Again, red bars compared to the blue. And what this means, to once again quote the authors, the data suggests that RGCs and EAE mice demonstrate dysregulation of proteins related to aging and senescence across disease time course. So once again, more indication of the aging relatedness of multiple sclerosis, or at least this model of it. Moving back again to human tissue, they found that markers of double strand breaks were higher in multiple sclerosis patients, as can be seen in I and J at the bottom here. So you can see that the red dots of the multiple sclerosis patients are on average higher than the controls. I'm not quite sure what the difference between these two figures are, though. Next, we move on to what, for me at least, is the exciting part of this paper. This is where the authors use cellular reprogramming to rejuvenate the retinal ganglion cells of EAE mice. And we'll look at this slide in more detail, starting with the study design in A in the top left. So 3 days before multiple sclerosis was induced in the mice, they were injected with 3 of the 4 Yamanaka factors, O, S, and K, using a viral vector, or AAV, which stands for adeno-associated virus. The injection was into the eye of the mice. There were also control mice, which were also given the viral vector, but without OSK. The motor skills of the mice were then monitored from day 7 post-injection until day 18, at which point the mice were sacrificed and the tissue was collected. Their visual sensitivity was also tested, I guess maybe these gray bars here. Looking at their motor scores, there was no significant difference between the OSK mice in blue and the control mice in black. Higher indicates worse. So this was to be expected, as the OSK was only injected into the eye and not into the motor neurons. Moving to the visual test in C here, mice were sort of placed on this platform and these rotating black lines went in front of their eyes. And if the mice reacted to that movement, it indicated that they could see something. And in terms of these colors here, so NR, the blue, means no response. And then I guess going from 0.3, 0.35 to 0.4 indicates a better response. And comparing the OSK to the control mice, the OSK mice had much better responses, as can be seen by greater purple here and greater green, and less red and blue compared to the controls. And judging by the star up top here, this reached statistical significance. Next, they looked at how the neurons fared. And starting with E here, which shows normal mice and EAE mice, which were not given OSK, you can see that the normal mice had much higher levels of retinal ganglial cells, or RGCs. For the normal and EAE mice, which were given OSK, there was basically no difference between the two. And comparing the EAE mice, either not given OSK or given OSK, you can see that the OSK given mice in light blue here had much higher levels of the RGC cells, indicating that they hadn't been killed off. Next, they looked at axonal swelling. And axons are the filaments which neurons use to transmit information to each other. And I guess they swell during multiple sclerosis. And you can see in gray are the mice which were not given OSK. And during the peak of multiple sclerosis, the axons swelled considerably, whereas for the mice given OSK, this swelling was much less. Next, they looked at the thickness of the dendrites. Dendrites are the filaments which receive information into the neurons. And here the OSK mice had much thicker dendrites in this cross section of the retina. This is better seen in the bar graph K here, where the OSK mice had a thickness comparable to that of the control mice. The mice not given OSK had a much thinner dendritic layer. Next, looking at gene transcription repression in N here, the OSK mice had much less repression than the mice which weren't given OSK. And lastly, in terms of the nuclear envelope, the abnormality was much lower in the OSK mice compared to those mice not given OSK, as you can see here. So it does appear that cellular reprogramming rejuvenated the retinal ganglion cells and reduce some of the pathologies of multiple sclerosis, not to mention improving the vision of the mice, which is great to see, pun fully intended. Finally, they looked at gene expression changes between the OSK mice and the control mice. Now, the experimental setup was pretty much the same. They were given OSK 3 days before induction of multiple sclerosis, then they were followed for 18 days before being sampled. Once again, there were no major differences in the motor score. However, there were major differences when looking at enrichment and the genetic sort of heat map. And won't go into too many details here, but just to show you how visually there is quite considerably a difference. So at the bottom here, you have naive or control 3-month-old mice, the EAE mice given OSK, and the EAE mice, which had symptoms of the multiple sclerosis. And just looking at the colors here, the OSK given mice were much closer to the 3-month-old young mice compared to the mice with symptoms. And just scrolling up here, you can see that when the colors are changed from blue to red, again, they're once again similar between the OSK mice in this column and the young 3-month-old mice in this column compared to the symptomatic mice in this column here. And on the right are all the myriad of genes being analyzed. And I'll finish off by once again quoting the authors. So these data suggest that OSK may revert gene expression changes occurring in retinal ganglial cells during EAE that alter processes related to neuronal function, nuclear integrity, autophagy, stress, and apoptosis, which is cell death. So it appears that on the genetic level as well, the OSK given mice reverted to a more sort of youthful profile. Finishing off with the graphical abstract, which basically just shows that OSK reduced neuroinflammation, increased the number of retinal ganglial cells, I guess these green dots compared to the fewer green dots down here, and also the fact that the mice, I guess, could see finer lines when these lines went across their faces after treatment compared to before. All in all, this was a very impressive paper, and I'm really excited to see this type of research taking place in Canada. This is the first time that I've come across a cellular reprogramming paper coming from our country. So if you know of any others, please let me know in the comments below this video. Although multiple sclerosis is not traditionally considered an aging disease, it's great to see the power of rejuvenation can also impact it. And hopefully we will soon see many more studies using cellular reprogramming, as it is one of the hottest fields in longevity research, and could have the potential to treat a myriad of other age-related diseases, and also hopefully extend our own longevity. And that marks the end of another longevity review. If you found it interesting, then please give us a like and subscribe to our channel. So until next month, Goodbye.